Okay, so this is chapter 10, part 1. And in chapter 10, we're not going to cover the whole chapter, just parts of it. We're going to look at cell reproduction, the cell cycle, and mitosis. Okay. No good spot for my video. So this figure is pretty cool. This is showing how a sea urchin goes from a single cell to 2 cells, and those 2 cells then divide to become this ball of cells, and then these cells continue to divide and then what we call differentiate to become the adult sea urchin, which is a type of echinoderm. So pretty cool pictures there. Those are electron micrographs, okay, and then this is just a photograph. So again, this is the process of mitosis. So this sea urchin is going from what we call a diploid zygote. And so a zygote is really just a fertilized egg. It's the first cell of that generation. And this term diploid, we're going to see this again and again. The term diploid means having a set of chromosomes from both parents. And so they have 2 copies of each chromosome. And so we are diploid, sea urchins are diploid, most animals are diploid, okay? And so again, we're going to hear that term a lot. So again, this was a zygote, right, diploid zygote, dividing into 2 cells. And then this again is the ball of cells, which is called the 16-cell stage. And then lastly, we have our multicellular sea urchin here. So mitosis occurs to produce more cells in an organism. And mitosis is a type of cell division, right? We're going to actually talk about 2 different types, mitosis being one of them. This process continues throughout life. And so inside of your bodies right now, mitosis is occurring in some of your cells. It's required for growth of an organism, for repair of tissue, and for maintenance. So, you know, mitosis is important to maintain homeostasis. We keep coming back to that same idea. Mitosis must be closely regulated. And so I'm not going to say too much here, but I do want you to know that when there are issues with mitosis, when there are cell cycle checkpoints that aren't met for whatever reason, that could lead to cancer. And so cancer is uncontrolled cell division. That's how a tumor forms. And then when you hear of metastatic cancer, one cell or a few cells from that tumor breaks loose and travels to a different part of the body and sets up residence there. Okay, so mitosis is a fundamental process. It's happening on us right now. It's important for growth, repair, and maintenance. But again, sometimes if it's not closely regulated, if there's mutations that occur that mess that up, it can lead to cancer. Okay, so I want to quickly show you, and actually this is one slide away, but I also want you to know that single-celled organisms, so prokaryotes, you've heard of that now, so organisms that belong to the domain bacteria or the archaea are prokaryotes, right? They have prokaryotic cells, cells that lack a nucleus. Mitosis essentially is how they divide. We don't call it mitosis, we call it binary fission, but it's basically the same idea. So the organism, its genome is copied, and then all of the other stuff in the cell gets copied, and they just kind of split the cellular contents as well as the DNA into 2 separate cells. And so again, that's how they reproduce. Not us, we reproduce differently. Okay, so let's look at the cell cycle. Okay, you're going to see this figure a lot throughout this whole chapter. And so it's good to get an idea of what this is. We're going to dive deep into the cell cycle in the next part of the chapter, but for now I just want to show it to you. So what is it? It's a sequence of stages or events in a cell's life where we go from one parent cell to 2 genetically identical daughter cells. So in a nutshell, that's what cell division is, right? We go from one parent to 2 daughter cells, and those 2 daughter cells are going to be genetically identical to each other and also genetically identical to the parent. Okay, so that's mitosis, cell cycle. The specific event of mitosis, so if we look at our cell cycle, really this is mitosis. Prophase, metaphase, anaphase, telophase, and then cytokinesis is really part of telophase. Okay, this is the actual stages of mitosis, and during mitosis, the nucleus divides, and then at the end here, the rest of the cell divides. Okay. So again, before we take a deep dive into this process, which we will, let's take a look first at genetic material. Okay. Okay. So again, I want to mention prokaryotic organisms. So again, bacteria and the archaea. Those are the two domains that have prokaryotic cells. And in this figure here, this is a bacillus, a rod-shaped bacterium. He's got a flagellum, and he's got pili and a capsule. And right here, this brown, looks like coiled stuff in here, this is DNA. Okay, so in prokaryotes, the DNA is not housed in a nucleus. Okay, but it is in a specific region called the nucleoid. Okay, so the DNA of a prokaryote or a eukaryote like us, so we call it our genetic material, or if we take into account all of the DNA in the organism, it's called the organism's genome. DNA is double-stranded. Whether we're looking at prokaryotes or eukaryotes, DNA is always going to be double-stranded. And again, in prokaryotic organisms, like the bacteria shown here, they only have one double-stranded DNA molecule, and their DNA is circular. Okay, and again, they only have one. It's found in a specific region of the cell called the nucleoid, not membrane bound, so no nucleus. And sometimes bacteria will also have little small circular pieces of DNA called plasmids. Not all bacteria have them, some do. Okay, so in bacteria, it's relatively simple. Okay, again, so a bacterial genome really consists of just one circular chromosome. Now us, eukaryotic organisms. So again, who falls into that category? All animals, including humans, all plants, fungi, and protists. And so eukaryotic organisms still have double-stranded DNA, but our DNA is not circular, it's linear, and there are usually several. So for example, let's look at humans. If you were to look at a human somatic cell, let me give you a definition here. So a somatic cell is a body cell. So any cell in our body except for our sex cells. Okay, so skin cells, cardiac muscle cells, brain cells, neurons, those are all somatic cells. What's not a somatic cell? Egg cells if you're a female, sperm cells if you're a male. Okay, so somatic cells again are also called body cells. So if we're looking in humans, inside of our body cells, our somatic cells, we have a total of 46 double-stranded linear chromosomes. Okay, our somatic cells are all diploid, or you might see 2N. Okay, we're going to be using these terms a lot. And so diploid, also represented by 2N, means that we have two copies of each chromosome. One originally from our mom, and the other set or the other copy from dad. And in a little while, I'm going to show you a karyotype. No, actually, I'm going to show you a karyotype. I can't remember where it was, a couple slides. And I'll actually show you what these chromosomes look like in a human. So again, we have two sets of chromosomes, or 46 total. Okay, because they're sets, we can also say we have 23 pairs, or we can use the term 23 homologous pairs. So again, you get one set of chromosomes originally from your mom, and one set of chromosomes originally from your dad, and those chromosomes are in every cell of your body. Okay, chromosome 1 from mom and chromosome 1 from dad are a homologous pair. They're the same size. Here's some of these words down here. So homologous pairs of chromosomes, same size, same shape, same location of centromere. They carry the same genes at the same genetic loci. The difference is that they may have the same allele or they may have different alleles. Okay, so say chromosome 1 carries the gene for eye color. Okay, we'll just say. Mom may have given you the allele that codes for brown eyes, and dad may have given you the allele that codes for blue eyes. Okay, or maybe both gave you the allele that codes for brown eyes. Okay, we'll talk more about this when we get into meiosis. So again, humans, as well as all other eukaryotic organisms, we have several double-stranded linear chromosomes. In humans, our somatic cells have 46 of these, or we can say 23 homologous pairs. If we were to look in our gametes, so sex cells, egg and sperm cells, not body cells, our gametes only have half that number. So that this term haploid means having half the genetic composition. Okay, and so in this case for humans, our gametes only have 23 total chromosomes, no homologous pairs. And so we can represent haploid with a 1N. Okay, so again, you want to get used to diploid, haploid, homologous. These are all very important terms. I just want to quickly mention fertilization. Again, we'll look at this again in the next chapter. Fertilization is when two gametes, so two haploid cells, come together to form a diploid zygote. And so each of these gametes is going to contribute one set of chromosomes. Okay, so you end up with the zygote having two sets. The zygote is the first diploid cell of the next generation. Okay, so in this figure here, I've just got a bunch of cells that are in different stages of mitosis, and you're going to actually get a chance to see a slide like this in the lab. Okay, so let me show you, this is a karyotype. And let me move my picture here. Okay, this is a karyotype taken from a human female somatic cell, so a body cell, not a gamete. What we can see here in this karyotype is we can see the chromosomes. Okay, and the chromosomes here are going to be dyed a specific color, right? Our chromosomes aren't all these beautiful colors naturally, but they're dyed so that we can see them. And they've been laid out next to each other so we can actually see the homologous pairs. And so you can see here, this is chromosome number 1. You have 2 of those, right? One originally came from your mom and one originally came from your dad. Okay. Chromosome pair number 2. So again, these are homologous chromosomes. 3 homologous chromosomes, so on and so forth until we get to 22. And then our last pair of chromosomes, chromosome pair number 23, are the sex chromosomes. And so for females, what's shown here are 2 X chromosomes. Males, this would be an X and a Y. Okay, so again, if you count these up, there's 46 total chromosomes here, or we could say 23 pairs of chromosomes. Okay, so this again is called a karyotype. It's a way that we can visualize all of the chromosomes in an organism's cell. And so before we move away from karyotype, I want to just make sure we understand the difference between maternal and paternal chromosomes. So if I just look at chromosome pair number 1 in this individual, this is a pair. Okay, I'm not sure which of these came from which parent, but let's just pretend that this first chromosome here came from mom originally. Then I would call this the maternal chromosome. And let's say this chromosome over here, the second one of pair number 1, came from dad. We would call this a paternal chromosome. And so again, you get a set of chromosomes from your mom and a set of chromosomes from your dad. 23 from mom, 23 from dad if we're talking about humans. Okay, other species will have different numbers. But for humans, which we have shown here in this karyotype, it would be 23. Okay, and so when we look at chromosome pair number 1, containing a maternal and a paternal chromosome, we call this a homologous pair. And so again, you've got 23 homologous pairs. So again, what makes them homologous? Well, they're the same size, the same shape, and they carry the same genes. But again, they could carry the same or different versions of the gene. That's what that term allele means. Make sure I define that. So the term allele means forms of a gene. Okay? And so with sexual reproduction, because you get genetic information from two dissimilar parents, or hopefully two unrelated parents, you end up with genetic variation, right? And so these alleles will interact in some way in you for expression, and you end up with genetic variation. So, you're not identical to either of your parents. You're also not identical to any of your siblings, unless you're an identical twin, okay? But your genetic makeup is going to be slightly different, which is going to give us genetic variation, okay? So some examples here, I already kind of mentioned eye color. You may get an allele that codes for brown eyes from your mom and an allele that codes for blue eyes from your dad, and you will end up with brown eyes, I should say pigmented eyes, not blue eyes. Blood type, same idea. If your mom is type AB and your dad is type O, you will end up with either type A blood or type B blood. Those are the only two possibilities there. Widow's peak, we're going to actually do some of these in the lab a little bit later in the semester. If both of your parents have a widow's peak, you may end up with a widow's peak depending on which allele they give you. Okay, so again, we're going to get into genetics in a couple of chapters, but those are just some examples of some variation. Okay, so we're going to end this chapter just talking about how DNA is compacted in our cells. So again, if we think about humans, so human DNA from just one of our somatic cells, if we were to kind of unwind it and stretch it out, it would be about 2 meters long, or 6.5 feet. That's one linear strand. Your cells are only about 10 micrometers in width. So that's a big difference, right? So DNA must be tightly packaged in order to stay organized and fit. And during the processes of cell division, in order to do that correctly without damaging or losing DNA. And so we have what are called chromosomes, which you've probably all heard of. So on the next figure, I'm going to show you kind of how chromosomes form. So before I actually do that, let me just tell you that in a non-dividing cell, a cell it's currently an interphase, okay? Your DNA is actually not too tightly compacted. It's spread out. It's still associated with proteins, but it's not tightly coiled, okay? And that's because if it was tightly coiled, then your genes wouldn't be accessible, and your genes need to be accessible for gene expression. So in a non-dividing cell, in an interphase cell, your DNA is only going to have the first level of condensing or folding, okay? And so if I look over here, this is a DNA double helix. And the first level of folding is your DNA is wrapped around these proteins called histone proteins. Okay, and so here's your DNA, and the histone proteins are shown in the middle. And there's actually multiple histone proteins involved. Okay, and what we end up with in this first level of folding is something called the nucleosome, which is also called beads on a string, because it literally looks like beads on a string under an electron micrograph. And so you can see here's histone proteins with DNA wrapped around them. Then there's a little space. And then here's another histone protein complex in a little space, and a histone protein complex in a little space. The spaces, where this is a little tiny stretch of DNA that's not wound around a histone, is called linker DNA. Okay, so the first level of folding is the nucleosome with, linker DNA that's kind of in between those histones. It's also known as beads on a string. And when your DNA gets coiled in this way, or condensed in this way, I should say, it's about 7 times shorter than it would be if it was just linear DNA without proteins. And we call that chromatin. Okay, so that's that first level of DNA condensing. And again, that's how DNA is going to be in an interphase cell. It's going to be in the form of chromatin, DNA with associated histone proteins. When your cells now enter into mitosis, the actual division part of the cell cycle, the DNA will need to get coiled or condensed smaller, okay? And so we go from chromatin eventually to chromosome, okay? So the second level of protein folding is coiling of the DNA. This involves other proteins into what we call the 30 nanometer chromatin fiber. And that's about 50 times shorter than a linear DNA without any protein. Okay, so you can see this here. There's this coiling of the DNA, and there's, again, a bunch of different proteins involved here in that coiling. And eventually, the third level is fibrous proteins pack chromatin. So we end up with, in this case, at the beginning of mitosis, a duplicated chromosome. And all of that chromatin is coiled tightly and, again, associated with proteins. Okay. At this level, the last level, the third level, this is going to ensure that chromosomes don't overlap. Okay, so this chromosome here, this is called a dyad chromosome. It has two arms, so one arm here and a second arm here. These two arms can also be called chromatids with a D at the end, and they're identical. Genetically completely 100% identical, these two arms. They're held together here by what's called the centromere. Okay. Because they're identical, we can also call them, again, sister chromatids. Each one is a single chromatid. Together, they're called sister chromatids. We'll get into that a little bit more as we get further in this chapter, and then when we get into meiosis in the next chapter as well. So that's it for the first part of chapter 10.